Hi everyone and welcome to the internet. What we're going to be doing in today's lesson is having a look at the process of exchange and transport. To start us off though, let's have a little look at some questions based around our previous lesson. So 4 questions are on your screen at the moment. Have a go at answering those in your books for me. So I'm just going to give you about 3 minutes to do that. So let's check those answers then. The first one, a stem cell is an undifferentiated cell. So give yourself a tick if you got that right. The 2 types of stem cell we've got are adult stem cells and embryonic stem cells. Give yourself a mark for each of those that you got right there. In plants, we refer to them as meristem cells. Tick that one. And then finally, the difference in terms of why we use one type of stem cell over the others is that embryonic stem cells, 1 mark, are used because they can differentiate into all cell types, mark for that bit, whereas adult stem cells can only differentiate into a limited number of cell types, and a mark for that bit. So maximum number of marks that we could get on our starter task today are 7, so give yourself a score out of 7. What we're going to be looking at today then, first of all, is this concept of surface area to volume ratio. So this is where biology and maths collide. So to start us off, let's see what we know from our math lessons. Write down the answer to these 2 questions. How do you calculate the surface area of a cube? And how do you calculate the volume of a cube? So I'm just going to give you about 30 seconds to get those 2 answers written in your books. Hopefully we said that when we're calculating the surface area for a cube, it's the length times the width to get the area of a single face, and then we multiply that by the number of faces, in that case 6. To work out the volume of a cube, it's length times width times height. So tick those if you got them right. If you didn't, I'd suggest you jot those down. I'll leave them up just for a few more seconds in case you need it. Let's put that into practice then. What I'd like you to do is calculate the surface area of a 2 centimeter dice and then calculate the volume of that same dice. So I'll give you just over a minute to have a go at this. So if we have a look at how to solve this then, our first part, calculate the surface area of a 2 centimeter dice. So the first thing we need to do is work out the area of a single face. So that is just 2 times 2, which is going to give us 4 centimeters squared. And then we need to multiply that by the number of faces. So it's going to be 4 times the 6 faces we've got on our dice, which gives us 24 centimeters squared. So that's the surface area of our dice. Then to calculate the volume, what we do is length times width times the height. So 2 times 2 times 2 gives us 8 centimeters cubed. Do obviously go careful to make sure you get your units right. So centimeters squared and centimeters cubed. So just go careful to get that right each time. Next question for you to have go answering then is, what is the surface area to volume ratio of the dice? So using the surface area and the volume you just calculated, work out the surface area to volume ratio for me and write that in your books. So to work out the surface area to volume ratio, we can start off just by writing down our surface area, which was 24 centimeters squared, and our volume, which was 8 centimeters cubed. So 24 to 8. But that's not in its simplest terms. So what we can then do is think what goes into both 8 and 24. And hopefully we've got the answer of 8. So 8 goes into 8 once, and 8 goes into 24 3 times. So what we've got as our surface area to volume ratio is 3 to 1. So now we've got an idea about surface area to volume ratio, we're going to move on to have a little look at these exchange surfaces and why it's important to understand surface area and volume ratios. Now, the first thing we're going to do is have a little look at this wonderful little creature called a tapeworm. So you've got a lovely big picture of it on your screen right now. What I want you to try to do is make me a bullet point list of any features that the tapeworm has to let it survive. So I'll just give you about a minute to have a go at jotting down any of those features. The ones I'm hoping you got written down are, first of all, the hooks and the suckers for attaching. So tapeworms are something that you'll consume if you eat things like undercooked pork. Now, what we've got, if you look at their head, which is the bit towards the top right corner of the picture, then there are the little circular bits, those are the suckers, and around the top you can see that there's these little hooky bits hanging off. Now, those are the hooks and suckers it's going to use to attach to your intestines. So it's going to embed the hooks into the wall, it's going to sucker onto the side there and get a really good grip so it doesn't get moved through your digestive system. The second feature that I'm hoping you got written down is that it's got a large surface area to volume ratio. And this is something that's really important when we're considering how organisms are able to get everything they need to survive. And the reason that that large surface area to volume ratio is really important to our tapeworms is because it means that the diffusion that they're going to use to get everything can happen fast enough. And what we find is diffusion distances are small. So that means rather than needing any kind of specialist exchange mechanism, they purely use diffusion to get everything they need from us. So jot down those two sentences at the top and then see if you can explain why that is important. I've just given you a big hint in what I've said there, so hopefully we can get that done. Hopefully the answer we wrote down is that it allows diffusion to happen fast enough to meet the demands of the tapeworm. So it doesn't need a specialist exchange surface, it doesn't need any kind of special transport systems. It's all about diffusion. If we now go on to consider slightly larger organisms, then we start to hit a problem with this surface area to volume ratio. Because as organisms get larger, that surface area to volume ratio gets smaller. Now, what we then find is that that's going to have a direct impact on what these organisms can do to get everything they need. So write down the top sentence and then have a go at answering the two questions underneath in your books. So the answer to our first question is that our larger organisms therefore can't use diffusion alone to get all those substances they need, because diffusion will be far too slow to actually meet their demands. Now, the reason that diffusion is too slow is because the surface area to volume ratio is too low. The way that our multicellular organisms have therefore developed to overcome this is that they've actually got specialized exchange surfaces. So they've obviously recognized the fact that in their evolution this isn't working with just diffusion alone, and we've then developed these specialist exchange surfaces to actually make it so that we can meet our demands. So make sure you've got those answers written down if you didn't get them right. Now, the reason these multicellular organisms have developed these exchange surfaces is to actually increase the surface area to volume ratio within certain areas of the body. Now, that means that they're then going to be able to take in the substances they need in order to meet their demands. So make sure you've got that top sentence written down, and then what I want you to try and do is think about how the lungs are specialised for gas exchange. And just write me an explanation of how we've got specialised gas exchange surfaces in our lungs. Hopefully one of the things we did manage to get here is that in the lungs we've got these structures called alveoli. Now that's one of the key things that we've got that makes our lungs a specialist exchange surface for gases. The whole purpose of these alveoli is that they're really folded to ensure they've got a large surface area. Their second key feature is that they are only a single cell thick on their outer surface, which means that there's a very short diffusion pathway. Third feature is that they've got a very good blood supply. If you look at the top right picture, you can see that surrounding them are these capillaries, and that means that we're going to be able to maintain the concentration gradient. So if we think back to the work we did on diffusion, we mentioned there are a few key factors that mean diffusion happens faster. We said a large surface area, got that. We talked about a short diffusion pathway, got that. And we talked about having a good or steep concentration gradient, and we've got that. So what we've got here is a very specialised area of the body, perfectly designed for gas exchange. So make sure you've got those 6 points written down if you've not already got them. The second region we're going to look at as an exchange surface are the intestines. So I've given you a picture that will hopefully help you out with this, but what I want you to do is, in your books, explain how the intestines are specialised for exchange of our digestive products. So I'll give you a couple of minutes to have a go. So the first one that we've got then is that they're folded into these structures called villi. These villi then have an additional structure on the surface called microvilli, and the whole purpose between those is to increase the surface area of your intestines dramatically. And the whole purpose of increasing surface area is so diffusion happens faster. So make sure you've got those 4 points written down, and you'll also notice in the diagram that we've talked about two other key features there. The first one being that they've got the 1 cell thick wall again, and the second one being that they've got a good blood supply with the capillaries closely associated to ensure that diffusion is going to be carrying on at a fast rate due to maintaining a steep concentration gradient. So get those points written down. If you want to do the sketch with the labels, by all means feel free. I'll leave this up for about another minute or so. Last bit we're going to look at today then is thinking about how we transport substances around animals and plants. So see if you can answer that question in your books for me. When it comes to animals, then we use the circulatory system to transport substances around the body. So give yourself a tick if you got that bit right. And plants also use a specialist transport system made up of two tissues, which you're going to learn a lot more about as we carry on through the biology course, which are xylem and phloem. So tick it if you got it right. If not, jot those points down. I'll leave them up for about another 30 seconds. Let's finish off today then with our plenary tasks. So what I'd like you to do is, if your target grade is up to a grade 5, it's the 2 columns on the left. If your target grade is over a grade 5, it's the 2 columns on the right. So I'll leave this up for about another 2 minutes. If you need longer, obviously just hit pause on the video. As always, you can find the answers to the plenary over on my website, sciencewright.weebly.com. You'll also be able to find that under the Internet tab. Just select the week that we're in, and then you'll be able to find things like the lesson PowerPoints, the answers to the plenary, any additional work, and if there's any little experiments I thought you could do at home, then they're there too. So have a look and get using that.